At this time, I'd like to introduce Scott Taylor. Over the years, DFO has awarded grants to several of Dr. Taylor's mentees, two of which we'll be presenting tonight. Dr. Taylor creates and teaches new courses, mentors multiple graduate students and postdocs, writes grants and manuscripts. Need I say more? What else can you find challenging? Scott, would you like to say a few words or introduce our guests?
Yes, of course. Thanks, Dave. So thanks everyone for coming. It's a huge pleasure to be able to share some of the work that students in my lab have been conducting, thanks to the generosity of the DFO research grants for students. So I'm first going to introduce Cori Carver. So Cori is an undergraduate student at the University of Colorado Boulder in her junior year. She's a double major in ecology and evolutionary biology, as well as computer science. And she's been working in our lab for how long now? Quite a while, since your first year, right? Since freshman year, working on the Boulder Chickadee study. And so she's really excited to be able to share some of her findings about chickadee diets by sequencing their poop. So without further ado, I'll turn it over to Cori for the first presentation of the evening.
Thank you, Scott. I will go ahead and share my screen. Good evening, everyone. My name is Cori, and I'm an undergraduate researcher at the University of Colorado Boulder. Thank you all for logging in tonight, and I'm really excited to share with you everything I've learned about nestling diet composition in chickadees. So humans are changing the natural environment around them at an unprecedented rate. This map, produced from the Last of the Wild dataset in 2008, utilizes different metrics of human development, including the absence or presence of roads, air and light pollution, and more, in order to get a feel on the most and least affected by humans, by the land most and least affected by humans across the US. As we would expect, most of the East Coast has quite high human footprint indices. As we move farther west, however, it appears that human impact decreases, except for one area that we have all come to know and love, Denver and the accompanying Front Range. While urbanization can have quite large impacts on habitat amount and quality, it can also change the way that different species interact together. For example, when human development begins, habitats can quickly become fragmented. And as development continues, things like native trees can actively be removed and later replaced with ornamental or fruit-bearing non-native trees in residential yards. Because these trees and the insects and arthropods that live in our areas have such tightly, are so tightly related through long evolutionary histories, when these trees are replaced, arthropods that have relied on them for thousands of years are forced to migrate or immigrate or go extinct, causing the insectivorous backyard birds that we have all come to love issues when they start to provision their nestlings because they feed their young insects. Non-native trees are important because they are able to support arthropods of great diversity and abundance. Non-native trees, however, are only able to support mostly predatory and generalist species. One study performed in the eastern US by Narango et al. In 2018 found that non-native plants played such a large role in how Carolina chickadees provision for their nestlings that indeed when non-native plant introduction increased, which we can see here along the x-axis, the population's ability of the Carolina chickadees population's ability to remain constant decreased. with R representing the population's growth rate. So as non-native plants were introduced in higher and higher proportions, Carolina chickadees failed to be able to continue to grow in numbers. So as Scott mentioned, for the last 3 years, I've been working with the Boulder Chickadee Study, which is a long-term study that is interested in understanding how these two birds, we have the black-capped chickadee on top and the mountain chickadee on bottom, have started to interact in Boulder County. The study is situated along an elevational gradient from Boulder, Colorado, including the University of Colorado Boulder, up to our mid-elevation sites along Sugarloaf, to the study's terminus at the University of Colorado Boulder's Mountain Research Station. Over 350 nesting boxes have been deployed at each of these 3 sites. And for the last 2 years, we have been monitoring all bird nesting events occurring in these boxes, from when nest building began to when the first egg was laid to hatching. Broadly, I'm interested in using the Boulder Chickadee Study framework to kind of get at this question of how non-native tree introduction alters the diet and breeding success of black-capped and mountain chickadees here in Boulder, Colorado. We have some insight into how other chickadee species are affected by the introduction of non-native trees, but we're specifically interested in how the ecology of life here in Boulder will change as more urbanization occurs and along the elevational gradient of the study. So to start kind of teasing apart this question and starting to learn more about potential answers, we first are interested in what chickadee nestlings are eating. So what are the adult chickadees provisioning to their chicks? We know for the 12 days that chicks are in the nest before they fledge, adults make multiple trips every day, bringing them back insect prey. So what are chickadees being fed? The second question is, how does nestling diet vary in areas of non-native versus native vegetation? So once we figured out what they're eating, we can start to look at the microclimate around each of these boxes to better understand if variation exists, and if it does exist, how is it different between native and non-native areas? And our final question is, so if this variance is occurring, does it correspond to increased nestling condition or their ability to fledge? So is the presence or lack of specific arthropod species, which is controlled by the type of trees present, control whether or not a nestling is likely or more likely to fledge? So for the last 2 years, I've been collecting many different types of data to try to get at this question a little bit better. One of those types is microclimate data for all of our boxes. So every time we deploy a box, we'll collect microclimate data about the surrounding area, including the type of tree the box is deployed in, the immediate climate, how high the box is deployed in the tree, and more information about tree species surrounding it. I've also conducted arthropod surveys, which consisted of me walking transects around our occupied nest boxes and using a canvas catch net to survey arthropods by dislodging them from trees and then storing them and later identifying them to order in the lab. Through the Boulder Chickadee Study, we've also collected reproductive success data for every pair of birds that has nested in our boxes. So we have a good idea of this basic breeding phenology data, including the lay date, the timing of hatching, how many chicks were successfully able to hatch, and how many were able to successfully fledge. We also have nestling condition and banding data. So after these chicks hatch And after they reach a certain age, we then head to the nesting box and we do some preliminary measurements where the chicks are extracted from the box. We weigh them, measure them, and take a small blood sample while we band them with a federal metal band. This allows us to be able to get a good idea of what chicks are likely to fledge, because we band them at a time that is relatively close to the date they're going to fledge. So if they make it up to that day, we're pretty confident, if nothing else happens, that they'll be able to successfully rejoin the population. I've also collected nestling fecal samples. So during the time when we would take nestlings out in order to weigh them and get some preliminary data, we would also opportunistically collect fecal samples from each of the birds. So depending on the situation, we would sometimes collect 1, 2, or more fecal samples from each nest, depending on kind of how that handling event went. And finally, we've also collected provisioning data through the Boulder Chickadee study, which represents kind of a way for us to double check or get a better idea of what nestlings are being provisioned, because fecal samples kind of act as a snapshot in time of what chickadees are being fed. But if we are able to get a better idea of what they're provisioning over time, then we can kind of double check ourselves and make sure that if these two, the fecal sample and the provisioning data, aren't matching up, we can investigate a little bit more. So here is a map of the fecal samples collected in 2019. So just to orientate us on this map, we have Boulder proper over here on the right. And then we have our boxes up Boulder Canyon, meeting us at Sugarloaf here. And you can see we have a nice collection from both Upper and Lower Sugarloaf. And then between Ward and Nederland, we have the Mountain Research Station. So samples collected from blackcap nestlings are represented in maroon, and we collected 16 of those in 2019. And samples collected from mountains are representative in navy. And as you can see, something interesting about this study is the fact that we were able to collect fecal samples from mountains and blackcaps in areas where we were excited to have them nesting. For example, while mountains usually remain at higher elevations, we had a few mountain nests in Boulder and were able to collect fecal samples from those nestlings. Similar to that of this lone fecal sample we were able to collect in Lower Sugarloaf, which was a blackcap nesting at higher elevations. So here's a great representation of what fecal sample collection would look like. So when we remove these cute little birds from the nest, we'll weigh them and then opportunistically collect fecal samples from them. So here's a snapshot into what the diet composition looked like in 2019. So this data is representative of the 16 blackcap nestling fecal samples that we collected in 2019. And again, when we start to look at this data, it's important to remember that these fecal samples tell a small part of a bigger story. So while this is representative of maybe what that blackcap ate that day, it's important to look over time on how these might change. And our 2020 samples will provide insight into that as well. So the majority or a large portion of blackcap chickadee diet is made up of butterflies and moths, which is what we would predict based on the gleaning behavior of blackcap chickadees, in which they actively hunt by turning over leaves and pulling caterpillars off of the back. Another large portion of blackcap diet is composed of flies, and it's closely followed by both true bugs and wasps. When we start to compare what we found through DNA metabarcoding analysis in blackcap chickadee diet to what we found in mountain chickadee diet, we can quickly start to see some pretty big differences. So while butterflies and moths still made up a large part of their diet, 34%, flies made up a much larger portion of mountain chickadee diet than they did in blackcaps. True bugs also played a much larger role in mountain chickadee diet than they did in blackcap diet. And as you can see, the variability in blackcap chickadee diet is much larger than that of the mountain chickadee. And while that data is very interesting to look at by itself, it's even more interesting to start to look at in comparison to the insects that I was able to collect using the arthropod surveys. So this data is representative of the insects I collected in 2019 around the same nesting boxes that the fecal samples were collected in. So this allows us to get a good idea of the arthropod populations that were present in these areas. So then when we start to look at diet information, we can compare what chickadees are utilizing versus what's available. So here across the x-axis, we have each of our 3 main sites. So we have Boulder, Sugarloaf, and MRS, each increasing in elevation as we go to the right. And along the y-axis here, we have proportion of diet, or excuse me, we have proportion of catch. So in Boulder, a very large proportion, almost more than 25% of our catch was true bugs, followed by spiders, beetles, and flies. When we moved to Sugar, we started to catch a lot more spiders, followed by true bugs. And as we move up in elevation to the MRS, we still continue to catch a large amount of spiders, an even larger amount of beetles, and the largest amount of flies of any site. And what's really interesting is when we start to compare this to the blackcap and mountain chickadee diet information that we were able to discover from the fecal samples. So up here on the left, we can see again the 2 diet proportions figures from the last slide. And now we're able to compare those to the arthropods that we were collected and counted by hand. So what's interesting is to start looking at what a large percentage of Lepidopteran, so butterflies and moths, made up each diet, but how it would look that most, so blackcap chickadee nestlings and mountain chickadees are both being fed. They're utilizing a large amount of what's available to them when it comes to Lepidopterans, because we didn't catch very many of, our catch was small at both Boulder, Sugar, and the MRS when it comes to Lepidopterans, and yet butterflies and moths make up a large proportion of both blackcap and mountain diets. So adults are utilizing much of the available resources when it comes to butterflies and moths. And then when we start to look at flies, we can see that mountain chickadees, which usually occur at higher elevations, seem to be utilizing a fair amount of the flies at Sugarloaf and at the MRS, while blackcap chickadees seem to be utilizing a fair amount of the available flies located at lower elevations at both Sugar and Boulder. So when we start to think about these big questions, like how does non-native tree introduction alter the diet and the breeding success of black-capped and mountain chickadees, a lot of these questions are difficult to answer with 1 or 2 pieces of data. So my project has really been focused on trying to look at as many different aspects of this question as possible, because it's definitely difficult to detangle some of these signals. And the diet composition information is a great example of that. So we know that the introduction of non-native trees can alter food webs on a very local level. And we know that fecal samples from insectivorous birds can provide insight into how local birds are utilizing changing arthropod prey populations. As we continue we know that breeding phenology data can reveal how changes in nestling diet are directly affecting specific bird populations. As I continue to work as an undergraduate researcher, every day I learn more and more, and my research has greatly contributed to my experience in college and my experience as an undergraduate. I especially want to thank everyone at the Boulder Chickadee Study and at the Taylor Lab for always being there and always providing me with guidance. I am also especially thankful to organizations like the DFO that have allowed me to continue researching and continue asking these really big questions in Boulder that I am very excited to continue researching. I think now we can open it up for questions.
Awesome. Thanks so much, Cori, for sharing some of your preliminary data. And just to let everyone know, so Cori will be continuing and sequencing last year's sample of fecal samples to add to this study. And so your first question, Cori, is when you say moths and butterflies are a major part of chickadee diet, you mean they're caterpillars, right? Not the adults?
Yes, correct. So the way that both blackcaps and mountains provision or hunt for their nestlings is by hopping from branch to branch and gleaning, turning over leaves, looking for caterpillars, or looking for caterpillars in like tree bark. So caterpillars in the literature play a really large role in how these two species provision their young.
Awesome, thanks. You have another question about how you actually collect fecal samples. Are you just picking up the droppings, or do you somehow remove them from the chicks?
Sure. So this was kind of a good learning process for us. We discovered that usually the easiest way is when we pick up the chicks to actually weigh them is usually when they'll defecate, and then we'll use a clean swab to then store the sample in a tube for later sequencing.
Awesome, thanks. And there's a question from Polly Reitz. Did you count or analyze the number of non-native trees in the chickadees' habitats?
Yes, that was part of our microclimate surveys and also part of our arthropod surveys. So in each tree that I collected arthropods from, I classified to species. And because non-native versus native is a really difficult kind of distinction to make, especially here in Colorado, where, for example, apple trees have an interesting classification in native versus non-native. I'm still working to kind of classify these species, but everything has been classified as species.
Yeah, and so that'll eventually get tied into trying to understand patterns in chickadee, like chick quality and such as well. So you have a question about whether chicks are unable to digest seeds.
I know during chick rearing, they mainly focus on feeding them insects. Scott, if you can give some more insight into that. I think arthropods are the majority of their diet during the time when they're growing up.
Yep. Yeah. So the adult chickadees, although they themselves eat a lot of seeds, especially in the winter, chicks are provisioned with arthropods exclusively. And once the chicks leave the nest, they can consume seeds, but it's not not typically provided to them in the nest yet. Awesome. Okay. So why don't we have one more? So we have one more question, whether we've considered putting nest monitoring data into Cornell's NestWatch database. Yeah, so we haven't yet. I'm just going to answer this one. I was a postdoc at Cornell, and so I'm pretty close with all the NestWatch folks, and we haven't figured out yet how we're going to coordinate them with them, but we are going to. So yeah, thanks for that, Kevin. And then we have, okay, one more question, and then we got to flip it to Erik. It's about how you identify the poop contents, by DNA or what?
Sure. Yeah, after the samples are all collected, they're sent off to a local company that runs analyses on them using metabarcoding, which is basically a technique that finds really small strands of prey DNA in the samples and then sequences them to order or even to species. So it kind of depends on the quality of the sample for us to kind of how much information we get from it. So usually most of the samples that had small pieces of DNA, we were able to get down to even the species, but we at least get down to the order to get a good idea of what they're eating.
Awesome. Thanks so much, Cori. So now we're going to switch focus to redpoll finches. And I know if we were in the room together, you'd get a raucous applause. So imagine that's happening right now. So now we'll switch to Erik Funk. And Erik is a graduate student in my lab. He's a fourth-year PhD student who studies two very exciting groups of birds, the rosy finches, which he was pictured with in the ad for today's talk, as well as redpoll finches, trying to look at the evolutionary histories of these groups as well as their plumage variation. And so Erik has some really exciting findings to share with you about redpolls. So I'll turn it over to Erik.
Yeah, thanks, Scott. And thanks to all of you at the DFO for having us out this evening. I'm really excited to get the chance to share with you some of this latest work that we've been doing on redpolls and some of their genetics. So I know this is a pretty bird savvy group, and I'm willing to bet that there are some redpoll experts even in the audience with us here tonight. But I want to start by just acknowledging that redpolls are a difficult group of birds to understand. And I don't just mean, trying to identify redpolls, although that's true, but just in trying to understand their biology and their evolutionary history. Our current classification of redpolls, though, includes 3 defined species, which includes the lesser redpoll, the common redpoll, and the hoary redpoll. And we can make some distinctions between these 3 types based on some differences in appearance. So for one, we can take a look at the amount of brown streaking that occurs on the sides of these birds. So you can see that in this hoary redpoll that I've circled over here on the side, there's kind of some whiter coloration to it, whereas in the common and lesser, it's a much more kind of heavily brown and streaked feathering. There's also some noted differences in bill size and shape, where houries have this kind of smaller, more pointed bill compared to the kind of broader and more conical bill of say the common. And then finally, the extent of red coloration. So kind of the extent and size of this red crown patch on the top and the kind of extent of this reddish wash across some of the breast feathers here. And so while the sort of extremes of these 3 types are likely easier to identify out, this variation is fairly continuous. And so there's lots of intermediate birds that make this somewhat challenging. Redpolls themselves as a group are also really widely distributed. So this is a range map from all of the redpolls. And so you can see that they're distributed across the whole Arctic, but they do show some latitudinal differences. And so in general, we see hoary redpolls being distributed at slightly higher latitudes than we see, say, the common redpolls. So in addition to having some difficult identification to them, redpolls are also difficult to understand because previous studies haven't found any genetic differences that easily separate out these 3 species. And so the most recent genetic study was one that was done in 2015 by my advisor, Scott Taylor, along with collaborator Nicholas Mason. And so they looked at 20,000 variable positions from across the genome of redpolls. And so this is a plot that I'm going to show a couple of these. So I want to take just a second to walk you through how to interpret them. This is called a structure plot. And each of these vertical bars represents a single individual redpoll. And what these analyses are trying to do is to try and examine how individuals group into clusters based on shared genetic variation. And so their assignment to any given cluster is indicated by the proportion of the bar that's colored a given shade. So in this case, you can see that all of the individuals that were included in this study are almost entirely colored the same shade of gray here. And so this is indicating that there's no real genetic variation that's separating out any of these individual redpolls into separate clusters. So we could look at this in another way as well, using what we call a species tree. And so these diagrams demonstrate the relationships of individuals to one another, or in this case, taxa. And when we look at these species trees, we don't really see any consistent pattern across these 20,000 variable positions to suggest any sort of consistent relationship here. And so this study, really is not that old. It was 2015, but even in just the last couple of years, there's been a really big increase in sequencing technology. And so while this study, was cutting edge in 2015, where it was able to sequence these 20,000 variable positions from across the genome, with access to improved sequencing technology, we can actually now sequence entire genomes of these birds. So we were interested in asking this question, what can we learn about redpolls by sequencing whole genomes? And we had a couple of questions going into this project. One of them was we were interested in if variation from across this whole genome was consistent with this previous genetic study in not really being able to detect variation that separates out these three taxa. With whole genome sequencing, we get much higher resolution of differences between birds. And so we are also interested in asking some additional questions like, can we determine the genetic variation that is perhaps responsible for some of these plumage color and bill shape variations that we see? And then finally, based on these differences, what can we kind of conclude about the evolution of redpolls? What can this trait variation and this genome sequencing tell us about redpoll evolution? So I'm going to use these three questions to kind of guide our talk today, and I'll revisit each one of these and put an answer on each one. And so we started by sequencing 73 whole genomes from redpolls across the distribution. So I'm going to show that same map again, but put some dots on it this time. And so points that are colored in blue are where we have obtained redpoll, hoary redpoll samples. Points in this kind of gold color indicate where we've gotten common redpoll samples. And then the points in this kind of aqua blue-green color are lesser redpolls. So from this map, you can see that we've got a nice broad sampling distribution across the whole of the redpoll distribution, including some key sites along Greenland and Iceland. So with large whole genome data sets like this, it can be really challenging to try and understand all of the variation that exists. So oftentimes the first step that we're interested in taking is trying to understand how individuals kind of cluster on an average difference level. So based on all of the variation across the genome, if we average it, which individuals are most similar to each other and which are most different. And so when we ran these clustering analyses, we found some genetic structuring or genetic clustering of redpoll individuals, but we found that it poorly aligned with both taxonomy and geography. So I'm going to put two more of these structure plots on the screen here. The plot on top is when we assign individuals to one of two clusters. And this bottom plot here is when we assign individuals to one of three clusters. And so you can see that kind of as a broad pattern, there's maybe a majority of these lesser and common redpoll individuals assigned to one, while perhaps the majority of hoary redpolls are assigned to the other. But it is not super well aligned with taxonomy in that there are individuals from each of these taxa that are kind of falling out in the other cluster. And in fact, when we add a third cluster, kind of expecting that it would perhaps pull out lesser redpolls, it actually doesn't really provide us with any additional information there, but instead is sort of equally putting all of these individuals into that third cluster. Another way of looking at clustering is rather than assigning individuals into a predetermined number of groups, we could instead let individuals kind of vary in space based on their genetic similarity with other individuals. So this is called a principal component analysis, and it's placing individuals more closely to individuals that are more genetically similar to it on average. And so again, we can see that there's some clustering taking place here, but it's not very, it's not divided by taxonomy in that we're getting clean clusters with just a particular species of redpoll here. And so it's really is interesting to start to think about what sort of variation in the genome is causing this pattern. And so when we generated this principal component analysis here, or this PCA, we were using all possible variation that we could detect in the genome. But some of this variation is really only found in maybe just 2 or 3 of the individuals that we sequenced. And so we were also interested in viewing what perhaps the more common variation was that's present in Redpolls. And so if we filter these data to include only the variation that is present in, say, 40% to 60% of the individuals, we could rerun our PCA and we get this, this plot here, where there's a much clearer definition to these clusters now. Again, it's not separating out individuals by any sort of taxonomy. And I'm not showing any of the sort of sampling data, but I will say that this is also not separating out by geography. So we don't see any distinct sampling localities falling out in a particular cluster. Instead, we're seeing the same sampling locality present in all 3 of these clusters. So from here, we were really interested in what is this variation that's present in these birds that's causing them to cluster in such an interesting pattern like this. Before I get into that question, I'm going to revisit our first one in trying to address if the variation that we detected from whole genomes agrees with previous genetic studies. And my answer to that is mostly yes. I do note that we detected this extra resolution, or with this extra resolution from whole genomes, we've been able to detect some interesting clustering. But my answer here is mostly yes, because it still seems to not necessarily align with taxonomy or geography. So in that respect, it seems that it's in agreement with the previous genetic studies that have been done on Redpolls. So knowing that, we can then kind of move on to our second question and trying to determine first, what is this genetic variation that is resulting in the clustering that we detect? And perhaps, is this the same variation that is maybe affecting the way that Redpolls look? So affecting these sort of plumage and bill traits that we outlined earlier. And so to try and get at this question, one of the ways that we looked at this was by taking all of the individuals from those different PCA clusters and comparing them to each other. Specifically, rather than looking at the kind of average genetic difference between these individuals, we were interested in how they vary across different regions of their whole genome. And so this plot that I'm showing here is what we call a Manhattan plot. And I'm going to walk through how to interpret this plot as well. This is just an example plot here, and then I'll show you the Redpoll plot next. But so each one of these little points that you see here is a specific position in the genome. And so you can imagine that we're kind of walking along a chromosome in the genome with each one of these positions being represented here. When you see the color change, this is just indicating that we are moving to a different chromosome. On the y-axis here, we just have a measure of how different the 2 clusters are that we're comparing. So at a given site, how different are these 2 clusters? If it's lower down on this axis, that means the 2 clusters are very similar to each other. Alternatively, if a point is very high, then this is indicating that these 2 clusters are very different from each other at that position. I can't remember if I just misspoke, but when it's low down, that means these are very similar to each other. Their genome variation is very similar. And so this plot gets its name, this Manhattan plot, for its sort of vague resemblance of a Manhattan skyline, where we've got lots of points mostly concentrated down here pretty low, kind of making up our horizon. And then we've got some kind of isolated regions that are moving up, forming the buildings of our Manhattan skyline here. This particular plot is fairly representative of what we might expect to see between 2 closely related species, where the majority of the points are pretty low down, showing that the majority of the genome is not very different between the 2 groups. But oftentimes we do see these sort of isolated regions where the 2 groups are quite different from one another, making up these peaks here. So just to remind you, this question that we were asking is comparing the individuals in the 2 outside groups of this principal component plot. So all of the individuals in this leftmost group compared to all the individuals in this rightmost group. And when we look at the Manhattan plot for that comparison, we see this pattern. And so right off the bat, you can see that it's pretty striking for a number of reasons. The first is that the majority of these points are extremely low, so very close to 0, indicating that there is essentially no genetic difference between the two clusters that we're comparing. Then, of course, there is an obvious single exception in this gray region right here. And so this is a region where there's a large amount of difference between these two clusters that we're comparing. And if we zoom in on this particular region and make a Manhattan plot just for that stretch, you can see that there's a very long stretch of DNA that has a large number of differences between these two clusters. So a large region of elevated difference. And this particular region actually is made up of about 55 million DNA base pairs long. So it's a very long stretch. And this particular stretch is located on the bird's chromosome number 1. So what this particular pattern is suggesting to us is actually something that's called a chromosomal inversion. And so I'm going to describe this on this little cartoon chromosome here. And so if you imagine that there's a DNA sequence running along this chromosome here, we might focus on just a particular stretch of this chromosome that I've highlighted here. And in this region, there's going to be a particular sequence of DNA base pairs here. And this sequence of base pairs are going to be in an order, and they're going to likely code for lots of genes that they themselves will be in a particular order. Now, when cells duplicate and chromosomes replicate, occasionally the physical DNA molecule that makes up a chromosome can actually itself physically break. And when this happens, the orientation of that broken part can sometimes get rearranged. Occasionally, this can do a complete 180-degree flip such that that broken region, when it gets reattached, is in the reverse order of what it was originally. And so this is where the name chromosomal inversion comes from. So now in that same region, we actually have the inverted DNA sequence, which means that all of the genes that are coded in that region are also now in sort of reverse order. So what this results in is 2 of the same chromosome that now is structured quite differently. And so I'm going to refer to these 2 inversion types as type A and type B. And so with this, if we revisit our principal component analysis plot, the 3 clusters that we're seeing here actually appear to be the 3 possible combinations of these inversion types. And so just like in humans, birds also have 2 copies of their chromosomes. And so in this leftmost cluster are the redpolls that have 2 A-type inversions. The rightmost cluster are birds with 2 of the B-type of the inversion. And then the central cluster are birds that have a combination of both an A-type and a B-type. So because it looks like variation between these 2 outside clusters is really only restricted to this chromosomal inversion region, we can kind of narrow in our questioning and focus just on that region and ask, are there genes within this region, within this inversion, whose functions are perhaps influencing redpoll evolution, and maybe specifically influencing the appearance of redpolls? And we do find that the inversion contains genes that influence appearance. And so the way that we got at this, or came to this conclusion, is we looked at the DNA sequence that underlied this inversion. So the stretch of DNA that itself was physically inverted. And we compared that sequence to the sequence of known genes, and then looked at their functions. And so if that function played a role in perhaps the development of a trait, we could then compare it to traits that differ in redpolls. And so as a reminder, what those traits were that we outlined in the beginning, is we noted that redpolls differ in their red coloration. So this is coming from a carotenoid pigment. They also differ in brown and black coloration, so primarily this brownish streaking along the flanks of these birds. And this is coming from a melanin pigment. And then we also noted that there's some differences in their bill shape and bill size. And so when we look at the genes that are located within this stretch of DNA, the names of these genes are likely not very meaningful. But when we look at their functions, we see that all of the the traits that differ in redpolls seem to have genes that are in some way related to their formation. In some cases, we even have genes that are perhaps controlling more than one of these traits. So this was a really interesting finding. And what this is indicating to us is that this region alone is perhaps responsible for likely the majority of these differences in appearance that we see in redpolls. As a way to kind of continue to look at the association of this inversion with appearance, we focused in on just a subset of the individuals. And specifically, these were individuals that were also included in that previous study back in 2015. And we focused on these birds because when a sample was taken from them, they were also measuring the physical appearance of these birds. So they were scaling, their color, they were measuring their bill and recording that data as well. So this particular plot is one that you can interpret just like we did with the PCA, where individuals that are more closely related to each other or are more similar to each other are placed closer together on this plot. On the y-axis, what we have here is a composite score that is related to its appearance. So lower down on this axis is a bird that overall is more hoary-like in its appearance. So it's much whiter in its plumage coloration, and its bill is likely a bit smaller and a bit more pointed. Whereas you move up to the top of this scale, you get a bird that's much more representative of a common redpoll. What's being shown across here on this x-axis is actually what we call gene expression. And so this isn't really comparing differences in the actual DNA sequence, but rather how that DNA sequence is being read by the individual, how it's being expressed. And so a really cool finding of this 2015 paper was that there's this really nice relationship here between how hoary-like or common-like a redpoll is and its gene expression data. So all I've done here is I've color-coded these points, these individual redpolls, based on the combination of the inversion type that they have. And so what we see is that birds that are more hoary-like in their appearance that are lower down here have 2 copies of this B inversion type. Birds in this upper corner, which are very representative of common redpolls, have 2 of the A type of inversion. And although we only had 1 individual with a mixed inversion type, it did fall out somewhat intermediate here in the middle of both of these axes. So this is suggesting to us that that this inversion type is in fact playing some sort of a role in generating differences in appearance. And as a way to sort of double check ourselves on this one, if our hypothesis is that this B type inversion is resulting in a bird that's a bit more hoary-like in appearance, then we might also expect the B type of this inversion to be distributed across higher latitudes. And so when we look at latitude, we do in fact see that as you move up in latitude, you see an increase in the frequency of this B inversion type. So in trying to determine the genetic variation associated with plumage color and bill variation, it does seem like we've been able to identify that variation, and specifically, it is almost entirely concentrated in just a single chromosomal inversion along the avian chromosome, number 1. So this is a really interesting result, identifying this chromosomal inversion. But now that we know this genetic variation that's associated with these differences, we were also really interested in what sort of a role it was playing in the evolution of redpolls a little bit more broadly. And so one of the first questions that we were interested in is, there's this large structural variation to one of these chromosomes, but is it playing a role in either acting as a barrier to reproduction or furthering any sort of divergence among lineages within redpolls? And our answer to that also is it doesn't look like it's playing any sort of role as a barrier. In the genetic signatures that we've looked at within these data, it indicates that gene flow between hoary and common and lesser redpolls is frequent. And so there were a couple signatures that we looked at to come to this conclusion. One of them was simply returning to this principal component plot and noting that there were a handful of individuals that had this sort of mixed inversion type here in this central cluster. And so if this was happening just kind of as a random chance, we might have only expected to, at the most, capture one of these individuals. But instead, this cluster made up almost 10% of our total amount of samples. So this was indicating that this sort of mixed inversion type individual was being produced somewhat regularly. And another way that we could look at this more directly is if we return to our Manhattan plot, we noted that one of the striking patterns of this plot is that the majority of these points are extremely low, indicating that there's no real genetic differences outside of this inversion region. And so we say that this pattern, we describe it as having low background genetic variation, meaning background to this single inversion. So this sort of a pattern could be due to gene flow. If you can imagine that if individuals from the two different clusters were exchanging genes, this would result in a genome that looks largely similar across the two groups. This pattern could also be produced if these two groups had diverged from one another, but only really recently. They may not have had enough time to accumulate sort of unique genetic variation that we would then detect here. So we did a specific test to try and compare these two hypotheses to one another. And this test did indicate to us some additional support for a hypothesis of gene flow among all of the Redpolls, rather than it just being recent divergence times. So we were able to note that gene flow seems to be occurring. If this is the case, then we were also interested in trying to understand all of these evolutionary processes that are perhaps in play within this Redpoll system that are working to keep both of these types of the inversion within the Redpoll population. And so one possible scenario as to how this inversion is being maintained is by some sort of a balance between natural selection, so some influence from the environment, and some assortative mating. So what I mean by assortative mating here is a bird mating with another bird that looks quite similar to itself. So an example would be like a common redpoll mating with another common redpoll. This is oftentimes what gets cited in the field. But I emphasized some here as we did detect frequent gene flow within this group, indicating that this isn't necessarily the only type of mating that's going on here. And so if the environment is playing some sort of a role, then it could be responsible for this pattern that we see in the frequency of this B type and latitude. And so if you remember, we mentioned that the B type of this inversion seems to be associated with producing a more hoary-like appearance to these birds. So whiter plumage overall with a smaller, kind of more pointed bill. And in particular, these two traits have been noted in other bird species as being likely adaptations to climates that are much drier and perhaps colder. And so this is indicating to us that there could be some local adaptation at play here that's resulting in these hoary appearing birds being distributed more northernly, and then the more common birds distributed more southernly. So it seems likely that this inversion is relating to local adaptation, but the detection of gene flow and the detection of all combinations of this inversion type is suggesting that it's not really playing any role in either generating reproductive isolation or in the further divergence of any of these Redpoll groups. And so in kind of revisiting the kind of broad evolution of Redpolls, the inversion type is in fact playing a role in generating variation in appearance, which itself is likely related to local adaptation, but not reproductive isolation or lineage divergence. So with that, I want to say a big thanks to all of my collaborators, especially my research advisor, Scott Taylor. The other collaborators on this project include Nicholas Mason at Louisiana State University, Snæbjørn Paulsen at the University of Iceland, Tomáš Albrecht at Charles University in Prague, and Jeff Johnson at the Wolf Creek Operating Foundation in Wyoming. I definitely want to give a big thanks to all of you at the Denver Field Ornithologists. These grants really go a long ways in helping grad students get research done. So thank you very much for having these grants, and thank you very much for inviting us to give talks tonight. Lastly, I want to give a big thanks to all the folks over at the Taylor Lab for being a great group to do research with. So with that, I'm going to leave you with a summary of some of the results. And then just wanted to say a final thanks to all of you who tuned into the talk tonight. Thanks.
Awesome. Thanks so much, Erik. And I'm sure if we were there together, there'd be an applause everyone could hear. So I know we're a little bit over time. I don't know, Dave, we have 4 questions in the chat. Can we go ahead and address those? Or yeah? Okay, so the first 2 questions are related, Erik. Are the 3 color morphs really separate species? And does your research support combining 3 species of redpoll into 1?
Yeah, so our research is supporting the redpolls acting more like a single species rather than 3. So it seems as though while this inversion is generating differences in appearance among redpolls, it's not doing too much to divide them genetically among as different lineages. And so the detection of gene flow across these groups is sort of supportive of the idea that they are behaving more like a single species rather than, say, 3.
Yeah, so there's some questions about gene flow as well. I'll just address them because I've been looking at them longer than you, Erik.
Sure, yeah.
So by gene flow, we mean movement of genetic material between individuals. So when 2 different individuals breed, they combine their genetic material, and Erik's results suggest that redpolls across the entire top of the world have gene flow occurring across populations and even across continents. So yeah, so the questions, that's the definition of gene flow. And so it seems like they interbreed where they co-occur. And then there's a question about whether the inversion occurs in other similar related species. And the answer to that is?
No, so it doesn't seem like it is present in other species. We did do some tests to try and see if perhaps this inversion was also found in species such as the crossbill. And I've also used some of our work on rosy finches to test it in them as well. And it seems as though this inversion is specific to redpolls. I will add, though, the presence of chromosomal inversions more generally does occur, though, in other species quite frequently, but not this specific one.
Awesome, thanks. So that exhausts the questions, and I'll turn it back over to Dave. Thanks so much, Erik.
Yeah, thank you, everybody.
Well, those were wonderful presentations, Cori and Erik. Thank you very much. We greatly appreciate your efforts and your contributions, and it's great having you tonight. It's great sharing with all of you.